For the first time, scientists from CNRS, Institute Curie and Sorbonne University, France, have been able to act physically on chromosomes in living cells. By subjecting the chromosomes to different forces using magnets, they discovered that chromosomes are in fact very fluid, almost liquid outside cell division phases. This study is published in the journal Science. This discovery was made possible thanks to the direct mechanical manipulation for the very first time of chromosomes in the nucleus of living cells. Until then, chromosomes, which are very long DNA molecules, were represented as being entangled like loose balls of yarn and forming a sort of gel. The observations reported in this new publication offer a very different image. Fluid and able to move freely, chromosomes are not restricted by other elements that make up the nucleus and are able to reorganize themselves. To reach these conclusions published in Science, scientists attached magnetic nanoparticles to a small portion of a chromosome in a living cell. Then they stretched the chromosome, exerting different degrees of force thanks to a micromagnet outside the cell. Using this approach, the teams managed to measure the response of a chromosome to external forces for the very first time in a living cell. Through these experiments, the scientists were able to see that the range of forces exerted naturally in the nucleus, for example by enzymes replicating DNA, is sufficient to substantially alter the conformation of a chromosome. This major discovery at the interface between physics and biology changes the previously established representation of chromosomes. It also adds new elements to our understanding of biological processes, the biophysics of chromosomes, and the organization of the genome.